In this video, we will talk about how neurotransmission at cholinergic neurons happen and cholinergic receptors. The cholinergic neuron, the preganglionic fibers terminating in the adrenal medulla, the autonomic ganglia, both parasympathetic and sympathetic, and the postganglionic fibers of the parasympathetic division use acetylcholine as a neurotransmitter. The postganglionic sympathetic division of sweat glands also uses acetylcholine. Neurotransmission in cholinergic neurons involves 5 sequential steps, which are synthesis, storage, release, binding of acetylcholine to a receptor and degradation of the neurotransmitter. (1) Synthesis. Choline is transported from the extracellular fluid into the cytoplasm of the cholinergic neuron by an energy-dependent carrier system that co-transports sodium. Choline acetyltransferase catalyzes the reaction of choline with acetyl coenzyme A to form acetylcholine in the cytosol. (2) Storage. Acetylcholine is packaged and stored into presynaptic vesicles by an active transport process coupled to the efflux of protons. Co-transmission from autonomic neurons is the rule, rather than the exception. This means that most synaptic vesicles contain the primary neurotransmitter, here, acetylcholine, as well as a co-transmitter that increases or decreases the effect of the primary neurotransmitter. 3. Release. When an action potential propagated by voltage-sensitive sodium channels arrives at a nerve ending, voltage-sensitive calcium channels on the presynaptic membrane open, causing an increase in the concentration of intracellular calcium. Elevated calcium levels promote the fusion of synaptic vesicles with the cell membrane, and the release of their contents into the synaptic space. This release can be blocked by botulinum toxin. (4) Binding of acetylcholine to a receptor. Acetylcholine released from the synaptic vesicles diffuses across the synaptic space and binds to postsynaptic receptors on the target cell, to presynaptic receptors on the membrane of the neuron that released the acetylcholine, or to other targeted presynaptic receptors. The postsynaptic cholinergic receptors on the surface of the effector organs are divided into 2 classes, muscarinic and nicotinic. Binding to a receptor leads to a biologic response within the cell, such as the initiation of a nerve impulse in a postganglionic fiber or activation of specific enzymes in effector cells, as mediated by second messenger molecules. 5. Degradation of the neurotransmitter in the synaptic cleft, that is, the space between the nerve endings and adjacent receptors located on nerves or effector organs. The signal at the postjunctional effector site is rapidly terminated because acetylcholinesterase cleaves acetylcholine to choline and acetate in the synaptic cleft. Cholinergic receptors. 2 families of cholineceptors, designated muscarinic and nicotinic receptors, can be distinguished from each other on the basis of their different affinities for agents that mimic the action of acetylcholine. Muscarinic receptors belong to the class of G protein-coupled receptors, metabotropic receptors. There are 5 subclasses of muscarinic receptors. These receptors are found on ganglia of the peripheral nervous system and on the autonomic effector organs, such as the heart, smooth muscle, brain, and exocrine glands. Although all 5 subtypes are found on neurons, M1 receptors are also found on gastric parietal cells, M2 receptors on cardiac cells and smooth muscle, and M3 receptors on the bladder, exocrine glands, and smooth muscle. Nicotinic receptors. It is composed of 5 subunits, and it functions as a ligand-gated ion channel. They are located in the CNS, the adrenal medulla, autonomic ganglia, and the neuromuscular junction in skeletal muscles. Thank you for watching. Please don't forget to subscribe to this channel